So I want to welcome everyone to join the virtual showcase on reproducible research in data science. I'm Jing Liu. I'm the Managing Director of the Michigan Institute for Data Science, and I'll be the moderator today. Because this showcase is a unique event, I want to give everybody a little bit of background. So MIDAS organized a reproducibility challenge earlier this year with the goal of promoting the good work of our colleagues and to showcase best practices and tools that our entire data science community can adopt. We received many wonderful submissions from across the campus, offering many different perspectives, including providing a definition of reproducibility, what can be and should be reproduced, making comprehensive documentation that allows for others to reproduce the results in a particular project, developing general platforms for coding or running analyses that standardize the methods for reproducible results across studies, metadata, tools, and processes to improve the robustness of results in the presence of variations in data, the computational environment, and human decisions, and methods to test the consistency of results from multiple projects. So the judges have picked a number of teams to put together this virtual showcase. I want to mention that it doesn't mean that the projects not chosen for the showcase are less good, but the projects featured in the showcase are particularly suitable for this format. I also want to mention that the videos and slides of the showcase will be online. We'll also build a resource collection online for interested people to check out what tools they can adopt. In addition, we will host a reproducibility day on September 14th. We'll announce winners of the reproducibility challenge at that time and who will receive cash awards. There will also be tutorials and panel discussions during the event. So today's presentation is has two speakers. William Wen is Associate Professor of Biostatistics. His research focuses on Bayesian model comparison, Bayesian multiple hypothesis testing, and probabilistic graphical models with applications in genetics and genomics. His co-presenter is his student, Zhao. And in this project, they provide a theoretical justification of defining the degree of reproducibility of high-throughput experiments with directional consistency and provide computational tools for such meta-analysis to assess reproducibility across studies. I want to mention that the judges of the reproducibility challenge liked the submission, particularly because it is a good example of treating reproducibility in a scholarly manner, yet with tools for practical purposes. So before I hand it over to the speakers, I want to remind the audience that you can put questions in Q&A or in chats, and we'll deal with this. We'll answer questions along the way. All right, so William and Yi, you can take away.
All right. Thanks everyone for coming to this webinar. It's a great pleasure to participate in this challenge. Let me just first share my screen. Okay, it's unclear. Okay. All right, can you see the presentation? Okay, great. So our entry to yeah.
Just now I can see, but right now it's just a black.
Right now it's
yeah.
It's good now or
no?
Oops.
Yeah, right now I see the the overview of the, but it's
okay.
not in a presentation mode.
Let me try again. Okay. How about now?
Still black?
Okay.
So if you go to the presentation mode and try to advance, would we be able to see anything?
Can you see something now?
Yeah.
Yes.
Okay, all right. So it's a problem like a full screen versus. I'll do this way. That's fine. It's less professional, but I think it works. So our entry to the challenge here is we're trying to consider the problem about quantification and controlling reproducibility in high throughput experiment. Particularly, I think this is the outline of the presentation. We'll get into the background of the introduction. So reproducibility in general refers to the consistency of results across studies amid answering the same scientific questions. In the previous showcase presentations, a few presenters do mention there is some distinction between the use of the terminology, for example, reproducibility versus replicability. So here, the problem we consider here is replicability. So reproducibility is sometimes narrowed down to if you can reproduce the computational results from the published work. And then replicability is a little bit tricky. So if there is something we don't have control over, for example, if you do an experiment investigating certain biological effect, and then if a different lab performed the similar experiment, replicability refers to the fact that the results are largely in line or concordant. But we will use reproducibility and then replicability interchangeably here. So one of our work is really motivated by this real life example. So we did a eQTL mapping with our collaborator, Matthew Simpson, who is also a co-author of this project. So the first batch of the data is generated using a microarray technology. So they measure the gene expressions and then trying to map the genetic variants that regulate the genes. And the time went on. Now the popular technology becomes RNA-seq. So if you're familiar with the molecular experiments, you know the RNA-seq is trying to get the read counts directly from the RNA molecules. So this is a it's a newer technology with higher precision. So the question is, we did the experiment twice with completely different technologies. Are the results concordant? So that's the fundamental question we're trying to answer here. But when we get into this, there's the situation that we need to consider. So this is exactly the situation we want to assess reproducibility, but we found there is lack of tool for us to essentially prevent us to do this straightforwardly. So that's kind of a motivating example, and then put the project into a application context. So to talk about the source of irreproducibility, we consider there are essentially two different factors that can come into play. One is usually in the high throughput experiment, there are always unwanted confounding and so-called batch effect. So usually when we do analysis, the existence of the confounding and the batch effect are not made aware of. So usually an analyst will ignore the existence of these batch effects. However, the consequence would be sometimes if the batch effect is correlated with your treatment labels, then there will be false positives. So we will show an example of this kind. The other source of irreproducibility, or is so-called, is just really the genuine biological heterogeneity. So essentially assessing the irreproducibility, the type of this type of the problem is not very different than assessing genuine biological heterogeneity. So think about if you test differentially express the genes in two different in two different tissues. So in tissue 1, you get a set of differential express the gene, and then in set 2, in tissue 2, you get a different set, most likely a different set of differentially express the gene, and then they are likely to be concordant. So this type of the irreproducibility is explained by genuine biological heterogeneity. So both of these type of the questions are important for their own purposes, but the first one is mostly the concern of reproducibility research. So we're not the first one to tackle this type of the problem. So almost all these, there's existing approaches using rank-based method to determine the reproducibility. So this is basically our baseline of our work. So they always assume some measures of signal-to-noise ratios. For example, the p-values to measure significance. Some of the key idea here will be reproducible signal, which is genuine and interesting biological signals should rank consistently higher in repetitions of experiments, right? So that makes sense. So if you identify something significant, you would expect in the reproducible results, the same set of genes also rank very high in your new experiments. So the representative approach of such kind are experiments rank test, and then this is the IDR is in brief for irreproducible discovery rate. It's by Li et al. So this paper is kind of a seminal work in reproducibility research, which is also kind of a foundation for ENCODE project for measuring chromatin accessibility in the ChIP-seq experiments. So given this existing work, why do we want to invent something new? So beyond the rank information, we realize there's many experiments offer natural, more natural and interpretable measurements of effect size. So this is usually in addition to rank information. So this kind of effect size usually have a directional interpretation. For example, if you have a differential expression experiment, you would interested in certain conditions, this gene is up expressed or down expressed. So this kind of a directional information is kind of critical for assessing reproducibility. So the fundamental question here is, can we actually utilize this additional information in quantify and control reproducibility? The next question we ask, can we quantify the variability of underlying effects based on observed data? So this is not really a hard problem to do. We have different ways, established ways in meta-analysis and in the statistical literature, general computational literature, in assessing heterogeneity. But we find what is really difficult here is the question, can we assess the extent of variability in repetitions of experiments? In the sense is how large or how significant or how variable of a signal or how inconsistent the observation should be that you call a particular measurement is irreproducible. So the assessment is always come from two different levels, right? So this is kind of you need to have a benchmark to define what is reproducible, what is irreproducible. And the assessment come into two different levels. One is the overall assessment for the repeated experiment. So when you perform an experiment, you need to know if there is a systematic problem with the experiment. For example, if the batch effect is in your experiment, it's not likely just impact the one single gene, it's likely to impact a bunch of genes. So the overall assessment will help you to identify this type of the systematic error, so to speak. And the second level of assessment is for individual experiment units. So people pay more attention to this in literature. So for example, you identify a gene that is associated with certain disease. And then the natural question is, in the follow-up study, in the validation study, are these results reproducible? So those are assessment for individual experimental units. So we're gonna deal with both of these assessments. So the most important thing in this talk is probably the next section, as we kind of propose an idea to quantify reproducibility with signed effects. we're trying to keep this, the talk non-technical as possible as we can. So the important thing is trying to explain our intuition and then how we come up with this, these key ideas. So as we said, we need to establish a baseline for classify reproducible and irreproducible signals. So we had quite a bit of debate on this, and then we come up with the final solution we take is this so-called direction consistency criterion. So this setup, so we call this in short just DC requirement or DC criterion. It's really a minimum requirement of the maximum tolerable heterogeneity for a common sense reproducible signal. So this is some sort of, in the decision theory, this is a kind of a following the minimax principle. So, so we all have different, if you want to quantify irreproducible, maybe everybody has a different threshold or different criterion. But at the end of the day, we need to pick a criterion that the whole community can accept. So this is the minimax type of the solution would help you in that regard. So the DC criterion is basically a very simple, very intuitive requirement. So we basically say with a very high probability, the underlying effects for reproducible signals are expected to have the same sign. So they are sign consistent. So if you see a signal that is upregulated gene, if you see a gene is upregulated in one experiment, if this signal is truly reproducible, you would expect that in the next experiment, you'll find the same gene is also upregulated. Actually, the fact that if you observe a downregulated a signal for the same gene in a different experiment, maybe is a strong evidence for irreproducibility. So that will be the consideration. So this is really trying to establishing a baseline for assessing the extent of heterogeneity. So a few remarks on this, the DC criterion and its implications. So we're talking about the consistency of the effects, but these effects is actually the true effect, unobserved the true effect versus you observe the effect. So the difference between the true effect and then the observed effect is you can view the observed effect as a contaminated true effect by the additional experimental noise, right? So it's really hard to define reproducibility based on the observed effect. To argue for this, you can imagine a hypothetical experiment. So you have a high quality gold standard experiment. So that's your experiment one. and then consider your repetition experiment. So if your repetition experiment is highly noisy, right, it's not, so essentially this repetition experiment will generate the observed effects maybe all over the place. So the point is the observed effects may not follow the DC criterion, right? But this should not be the standard we use to judge if the first experiment is reproducible or not. It's the reason the observed effects has inconsistent sign is because of the second experiment is too noisy. So therefore, we should consider the noise has an effect on observed effect. And then when we're trying to assess the reproducibility, we should be talking about the unobserved effects. And actually observe this actual noisy level is help us to kind of a pinpoint if we can pinpoint the true effects or not. So take consideration of the noise is kind of important. Secondly, this is it's an extension from Tukey's argument on detectable effects. So Tukey gave this general argument against testing null hypothesis, especially the sharp null hypothesis, saying, for example, in the differential gene experiment, so the general null hypothesis is the expression level doesn't change under different conditions. But that kind of a null hypothesis is silly. You don't believe they are exactly the same in two different conditions. Instead, Tukey argue the true effects, well, I mean, what we can do is we can identify those effects as large enough or strong enough that we can comfortably tell the sign or the direction. So the same idea apply to reproducible research. So this, we're just basically extend that idea. If you kind of consistently can tell the sign, then the direction of the effect, then that's a good evidence for reproducibility. The third one is because we're using a sign, this is to enjoy some scale-free property. So this is extremely important for our motivating example. Remember that the eQTL in the first study is measured by the microarray experiment, which use intensity to quantify the gene expressions, whereas in RNA-seq, these the count of the RNA abundance changes to read counts. So essentially, you cannot really compare intensity versus read counts, at least trivially. However, those kind of a caveat doesn't really prevent us to apply the DC criterion to assess the reproducibility in this context. Finally, I want to emphasize, although this is DC criterion is trying to emphasize kind of a one direction variability, right? If you have a true effect, then we kind of are saying you cannot cross the line being the neutral line 0. But in most of cases of the implementations, this defines a range. So not only you cannot cross the line, you cannot the reproducible signal cannot be too exaggerated on the other side of the true effect, right? So although this is using half of the variability range to define a kind of accessible variability, it's a really kind of give you a full range of accessible heterogeneity. So this comes important when we apply this to publication bias detection. All right, so moving on. So with this kind of definition of reproducibility versus non-reproducibility, we can actually have classifications of all signals in the high throughput experiments. So it will be 3 different classes. The first one is non-signals. So the non-signals are consistently 0 in all experiments. And then the second class is being the reproducible signals. So those signals are consistently non-zero in all experiments. And then the last category is a irreproducible signal. So those signals are inconsistent effects across experiments. So they are mutually exclusive. So that's the important thing. The other kind of, the other issue is rank-based method usually don't have this refined classification, right? Rank-based is basically consistently ranked and not consistently ranked. Here we use additional information on the effect size. Therefore, we can construct a more refined classification of all signals. So that's one of the advantage of this approach, just conceptually, versus the rank-based approach. All right, so I have to talk a little bit about the statistical modeling. How did we translate the DC criterion into a usable statistical modeling tool? So these are served as examples. It's just trying to showcase that how we translate or interpret the DC criterion. So the model we have here is, if you're familiar with meta-analysis, is very close to that context. So the beta ij hat is the observed effect. And then usually they also come with a delta ij, which is the standard error of the measurement. So as we said before, this is a quantification of the noise level. So given the true effect, then we usually make this sort of assumption. This is a normal assumption here. It's centered the true effect with the variability, and the observed effect is centered at the true effect, and then with the standard error defined by sigma ij. And now what we need to do here is trying to model the heterogeneity of effects across different studies or different experiments. So this maybe look a little bit strange, but this is really a consequence of the DC implication or the DC criterion. So this is a consequence of the probability statement we listed down here. So all the beta ij, so that's the true effects in different studies for the same unit I, has the correct sign as the beta I bar, which is the grant effect. So if you think about, there is some genuine true effect, and then they have, it's measured by all different experiments. So that's the beta I bar. So this function, this probability can be quantified by a phi function, phi over k. So phi is the cumulative distributional function of a standard Gaussian distribution. So the implication for this is if the k goes to 0, then 1 over k goes to infinity, the probability will goes to 1. So with probability 1, you will see all these betas have the same sign. And the other extreme, if the k goes to infinity, and then the 1 over k will goes to 0, then as you can imagine, so the beta ij has the same correct sign as beta I bar is essentially 50%. So the function will give you 0.5. But this is just one implementation of the DC requirement, DC criterion. We have another one, different model. So this model is essentially the meta-analysis model most people use. So there will be a heterogeneity parameter phi square used in place of k for the previous slides. So the next line is just trying to show by specify a different parameter r, so it's a function of phi and omega, we too can have a DC requirement here. So the beta ij have the correct sign will be computed from this complicated function, but this is only a function of r. All the others are maybe hard to compute analytically, but it's deterministic with respect to R nevertheless. So the key point here is when you actually do a meta-analysis, there is generally no guideline to select phi, right? So there is, so some of the guidelines tell you you need to estimate phi from the data. But here, what we're trying to do is we define a phi according to the probability here, with high probability being reproducible, then we can find a range of phi's that satisfy the reproducible requirement, and then we use that to do essentially Bayesian model comparison. So that's essentially the next slides. All right. So how do we use this this set of the tools or these concepts? So just very briefly to talk about this, what we could do is we kind of employ a Bayesian model comparison approach. So with the same observed data from repeated experiments, we can compute the likelihood or the marginal likelihood or base factor for each scenario. So we can evaluate if a data is generated from is generated for a null signal, or is for a reproducible signal or an irreproducible signal. So they can come up with, usually they come with a different likelihood or the base factors. So now you could do the comparison. But the high throughput experiment gave us the ability to estimate the proportion of each signal category by EM algorithm. So this is essentially, you can estimate the prior information from the data. So the idea is very simple. So thinking about every signal every signal or every experiment unit has this true indicator saying this is the data generated from either from the null, reproducible, or irreproducible signal. And then the indicator itself, you can consider is missing. And then if you have a missing data in this case, the natural way is to do the partial pooling and then trying to estimate those indicators back at the, as a kind of a byproduct. You also get the proportion estimate of the whole data. So we're going to use this estimated proportions as an assessment of the overall experimental quality. Right, so the resulting posterior probability with the prior and the likelihood, you can compute the posterior, and then the posterior probability is a natural way to do classification. And the added benefit is we can do the essentially the local FDR control for reproducible signals. So this is extremely important because most of cases we're really trying to control the false discovery rate for either the irreproducible signal or null signal. So this provides a fundamental way. If you can compute the posterior probability for reproducible signal, then you can automatically plug into this local FDR control procedure to achieve that goal. If you think about the alternative, if you're trying to set up some sort of hypothesis testing that will be very difficult. The reason for the difficulty is, in this case, you're trying to reject the null hypothesis and then trying to say something about, make a statement of a reproducible signal, but your null hypothesis is necessarily need to be compound with the two scenarios, the null scenario and then irreproducible scenario. To come up with a natural null hypothesis for this is not trivial. All right, so with this, we're just trying to show you a few illustrations of use cases. Some of these are real data, some of these are simulations. The first example we did is trying to detect the batch effects in high throughput experiment. So what happens here is we're going to simulate the data with and without batch effect for two experiments investigating the same thing. So the Y here, you can think about that's the gene expression. And the mu here is the intercepted term. The beta define the true biological effect. The xj is either case or control in our setup. So it's either case. The delta here is represent the magnitude of batch effect. The gamma j represent the batch effect, right? So for example, which samples are measured together as a, so that's essentially a batch label. Okay, so we have added noise sampled from a normal distribution. The delta ij is also kind of a sample from a normal distribution. And with the eta square, quantify the magnitude of the batch effect. So this model is commonly used in batch effect detection as a location and scale model. And then the batch effects are partially correlated with case control status. If it's not complete, if it's completely independent with the case control status, then this doesn't really introduce type 1 error. So the batch effect is just, you just lose power without considering them. However, if the batch effect is partially correlated with case control status, they will introduce false positives. So the idea is, well, I mean, this commonly occurred in real life research when you actually do all the cases in one batch for RNA-seq and then all the controls in one batch in RNA-seq. At least in early days of the practice, this is very common mistake. The batch labels are latent. It's not available to the analysts. So we're going to vary the magnitude of batch effects. And then we're trying to evaluate the replication data sets with respect to, the replication data sets is contaminated with the batch effects, but with different magnitude. And then we're trying to compare that to a gold standard data set, which is not contaminated with by the batch effect. So here is the graph showing you what happens here. So the pi IR represent these, so these red dots here represent inferred or estimated irreproducible proportion from the data. So with the pi r, the right line is the reproducible proportion from the data. And then the blue lines are using a different set of the method I presented earlier. So this is the CEF versus META. So the important thing is not comparing the two different methods. It's basically trying to see, so our method can effectively pick up the impact of the batch effect from certain, when the batch effect, the magnitude reaches certain threshold, like in the 0.6 and then 0.7 level. So it's intuitive. If the batch effect is too small or goes to 0, you probably cannot detect it. And they don't really have an effect to cause false positives. So we find that in this, at least for this type of the experiment, it caused the problem. Like you will see significant false rejections at the point when you have 0.8, like in this level. But in this level, I think our method, I think just by using the DC method, you can effectively pick up the signal and then basically make a claim that the data set is contaminated. So this simulation, the graph is obtained without any true biological signals. So any rejections from the data set will be considered false rejections because the simulation we used has the true biological effect that sets as 0. Okay, so we again simulate with a mixture of true biological signals. And then in this, the setup is more complicated. Half of the true effects are contaminated by the batch effect, half of them are not. But anyway, by applying the proposed approach, what we find is we can properly control the FDR in identifying reproducible signal. So when we say we control FDR at the 5% level, we meet that guarantee. In comparison, if you don't use this approach, most of your rejections will be irreproducible because as the magnitude of batch effect getting larger and larger. We also experiment using the available batch removal procedures before we apply, before and after, and then we can assess how effective of these available batch effect removal procedures. So the method we tested are COMBAT and SVA. So these are popular approaches used to supposedly remove the unobserved batch effects. So we find generally they are effective, but they don't really eliminate all the false positives, which is reasonable. I think the problem getting to remove the batch effect is extremely difficult here. So we also have a comparison with the rank-based method, specifically IDR. So there, I think the general conclusion is we find IDR can be difficult to apply in this case because of the computational difficulty. I think we just cannot get the method converged with reasonable time. So the second experiment we tested is more like is using the real data with concordance. We're trying to test the concordance of gene level genetic association testing. So the particular experiment we did is so-called transcriptome-wide association study of human heights. So those are, if you're familiar with genetic association studies, so those are just change the unit from individual genetic variants into the genes by grouping the relevant genetic variants together. So the grouping is done by annotating the relevant eQTLs. So, so I don't want to explain too much about the details for the TWAS. If you're interested, you can check them out. So we're essentially compared two large consortia, the results from the two very large consortia. The GIANT consortium, which is a comprehensive study of human traits, and height is one of those. So they have sample size with 250,000. And the recently made available, the UK Biobank data. So they also have height as one of the traits they investigated, but the sample size is much larger with 337,000. Those data are only made available through the Z statistics. So when you test each gene, they provide a Z statistic to you. And if you just compare the Z statistic across these two studies, you kind of get some sort of perception that they should be close, but not exactly the same because the sample size is very different. If you think that the actual effect size are similar, but the noise level is very different. So the Z statistic should, it's essentially a measure of signal to noise ratio. So the magnitude of these statistics are expected to be inconsistent. But can we say something about reproducibility? The GIANT, which is a much smaller study, reproduce the UK Biobank or the vice versa. If we do this, so before we show our result, so what's the available tools make some sort of assertion. So this is based on the Cochran's Q test. So if you're familiar with this, this is basically testing against the fixed effect assumption. That means the Z statistic has to be identical. So, well, however, the excessive of this small p-values indicating that abundance of genes doesn't really follow the fixed effect assumption. In our application, we find most of the genes are come from the majority of the genes are has no contribution to not associated with the heights. At least the now is 84%, but well, I mean, depends on your expectation. There's plenty of genes associated with human heights. The important thing is we find 16% of the genes are reproducible, and then only about one. So this is a proportion indicating most of the genes are indeed reproducible. So most of the significant findings are reproducible. So this is a biplot of the Z-scores from one study versus the other. So the green line here shows the diagonal line, because one study is significant, has a larger sample size. So you see this is a UK Biobank. Sorry, you cannot see the label. So this is the giant. So the UK Biobank tends to have larger sample size for the same gene. So nevertheless, I think the right dots indicating the individual level reproduce probability greater than 90%. So the method assert most of the, so at least in this is a decision boundary. So these boundaries saying after this decision, those are considered to be reproducible genes. And then if you see on this area where you have genes have generally different signs for the association, they are not considered to be reproducible. All right, so I would just want to briefly mention some extensions and the future work. So one of the things we start to considering when we start revising our manuscript is consider this application to publication bias. So publication bias refers to published results tend to be exaggerated versus the true effects, because when you only if you reported the significance findings, the paper tends to be published. Therefore, the name of publication bias. This is also known as winner's curse or p-hiking in different areas. So publication bias is often convoluted with a genuine biological heterogeneity, or there's just, so it's difficult, especially when you have limited studies, to determine if there is truly a publication bias or it's just the variation of the p-value, the significance levels are due to genuine heterogeneity, allowable heterogeneity. So available detection methods are like aggregate regression. So those relies on detecting funnel plot asymmetry, but requires multiple experiments. Typically, you need to more than 10. So why especially the DC criterion can be helpful here? Because by applying the DC criterion, we can decompose the acceptable heterogeneity and then the remaining, what we can call the residual heterogeneity. So that's the simple idea. If you decompose that, you find excessive residual heterogeneity. That residual part has to be interpreted with some sort of a publication bias or other unintended bias. So the benefit of this work is actually work with limited studies and does not really require high throughput setting. So we can consider this as a model comparison problem instead of thinking you have to have the EM algorithm to help you to estimate the proportions. anyway, so we did a proof of concept, the simulation. So the simulation is performed through the two different procedures. In the first procedure, the smaller study is naturally reported. There is no selection process. In the second batch of the study, we kind of simulate a very aggressive p-hiking behavior. So for the small study, only if your p-value is less than 0.01, we start reporting. Otherwise, we just keep simulating. So in that case, the second batch contains all of these publication bias. So what we can see here by using the model comparison approach, so that's so we can distinguish between with and then without publication bias with relatively high sensitivity and then specificity. So some future directions. So we can, well, for publication bias, we can actually combine the information of funnel plot asymmetry and the directional consistency principle, and then make it more powerful in detecting publication bias. And then the second one, I don't want to talk too much. This is related to the conceptual replications. So this is terminology used in probably originated from psychology research, but is actually covered quite a bit of applications, including our motivating example, the same experimental data measured by two different set of experiments. So there's some caveat to directly applying our approach to conceptual replications. So we're working on that to address all these caveats and then make it more powerful in general area of reproducibility or replicability research. So finally, I want to acknowledge our co-authors, Matthew Thompson. It's really, he did most of the heavy lifting in this project. And then due to the time and concern, I don't think, Yi, do you have anything to add?
No, everything is perfect.
Okay, thank you.
Yeah.
So we thank MIDAS for organizing this this challenge. If you're interested in our work, they are the preprint available at this address. So with that, thank you very much.
Yeah, so William, thanks so much for the wonderful presentation.
Thank you.
Yeah, so we have a few minutes for questions. To the audience, if you guys have any questions, please feel free to put them down in either the Q&A or the chat box. And while we see what questions come in from the audience, let me start by asking you a few questions about the implication and the application, really, of this. So the first question, I guess, is how would this influence the work of the practitioners, in a sense, right? The people who design and who run the high throughput experiments, either by working with biostatisticians like you or on their own. How can they use this concept to both compare the results and also to, you know, see how this inform their future study design?
Yes, I think that's an important question. So we consider the scenario is typically, at least for our current implementation, is you do have a reference data set out there, and then you want to ask the question if our new results are considered to be reproducible from that. So the part of the work we didn't represent is use this framework to detect the genuine biological heterogeneity. So for example, you have eQTLs get from the different tissues, from the lungs and then from the livers, and then you want to ask the question, what results are reproducible or irreproducible? So those, in this type of the situation, I think experimental design is easy, and then you just needed the tool, I think our two serve that purpose. For general kind of a batch effect, I think the application could be, so you may have either the kind of a reserve the data, or you have a reference data set, and then you want to evaluate if your kind of a batch 1 and then batch 2 are reproducible in that sense. But in any sense, I think this is, I think the general purpose is assess heterogeneity. I think how you use it depends on the applications, and then it depends on if you have a so-called gold standard, or you don't have a gold standard. You just want to make a relative comparison of multiple experiments. But in both cases, I think use this type of the method could be helpful.
So how high is the hurdle, though, for the practitioners to actually use this approach more in a more sort of, consistent way, I guess?
So you mean the computational cost, or?
Well, computational cost, and also in general, how much effort do they need to invest in, so that they can use it properly?
I understand. I think, so this is what we learned from the other showcases from this, the MIDAS challenge. So basically inspired by this, we actually created a Docker image, and then also have all these kind of simulations, and then the demonstrations of the real data analysis kind of bundled in. So we believe it's quite easy for other people to follow, but we are being subjective. But just trying to say, we learned a lot from this whole process. And I think we hope this whole thing should be easy to follow and easy to apply.
That's great. We would like to get your Docker image and include on our website collection.
Well, that would be great. Yes.
Yeah. Vera asked a question. William and E, great work. This is terrific. What level of data would you need from the studies to implement this framework? Also, how does one combat cross-study data processing and normalization that might impact downstream analysis?
Yes, I think that's a great question. So at the minimum level, we only need the estimate the effect size and then the standard error for each experimental unit, right? But you have usually, if your experimental unit is a gene, then that means thousands of genes. So we haven't used this for real data for in batch effect detection. So our simulation is pretty straightforward. I think we control everything being normalized, being properly analyzed, because I think it's mostly as a proof of concept, but that's definitely we're interested in to get into get our hands more dirty, and then get into the details of normalization and other pre-processing steps.
Let's see. So I don't see any other question. Let me ask one more question before we end this, which is there is a lot of sort of theoretical development on this type of methods, right, to assess the high throughput experiments. On the other hand, we all see publications where authors are very excited about, hey, we found 1 gene, or we found 3 genes that are so important. So from your point of view, is there a gap between the theory development, right, and their application to really guide the high throughput experiments in the results interpretation? And how do we improve?
Yes, I think, for example, in our settings, we always assume all the data are available. So as you said, sometimes people focus too much on that, the published or the significant data. I think that's a great point. I think from at least from our results, we need to, if you're trying to evaluate the overall quality of a study, you necessarily need to obtain those non-significant measurements and then do a better assessment for the overall study. Nevertheless, I think that the second thing is the people get excited about the single gene findings. I think this is also related to our discussion on the publication bias, right? So you find something, and then if you do experiment, you may not find that they are in line with each other. But your replication is actually serve as a comparison point to the published result. You may help, at least some of the method here maybe help to assess if either the published result suffer from the publication bias or there's something fundamentally, incorrect. Yes, I think that's a terrific question. I don't have a complete answer to that, but I acknowledge. I think one is all the data are important. Secondly, the significant results, you may be able to use a different set of the method, like publication, to treat as a publication bias detection analysis, let's say.
Yeah, so I don't see any other question coming in from the audience. And given time, let's end our presentation here and just assume that you heard wild applause for your presentation.
Thank you so much. Yeah. I appreciate everybody come here. And if you're interested, get in touch with us. And we're excited about this work as well. Thank you.
Oh, wait a minute. Are you still there?
Yes, I'm still here.
Yeah, well, Christopher Dampier just asked a question.
Yes.
Can the INTRIGUE framework be used to measure the variability in the underlying measurements, for example, gene counts, from one sequencing replicate to the next, as opposed to downstream association statistics?
So I don't think I have sufficient information to say one way or the other. I think we need this is just me. I think we need to learn a little bit more details. I think I welcome the discussion. I'm happy to discuss more, but I think for now I don't really fully understand the question. Sorry.
So Christopher, please feel free to contact William and E afterwards, or if you want to unmute yourself and have a discussion right here for a couple minutes, please do so.
Oh, cool, thanks. I didn't know I could unmute myself. Yeah, thanks. Okay, so the basis of my question is the concern that I have from time to time when we have an RNA sequencing experiment that we run, and we need to add extra depth, to get a better estimate of what the counts are in the genes. So we do another round of sequencing, and then another, and we keep doing them. And then,
yes.
A common practice is to simply concatenate all those reads. But I don't think that's necessarily safe to do. Sometimes, you want to sometimes it occurs to me like, oh, well, maybe there's a way that I could control for the variability, that technical variability from batch to batch in a formal way. And I was thinking that your framework sort of addresses it, but the examples you discuss are mostly focused on the downstream analysis, like, for example, an eQTL association.
That's right.
So I think that my use case is maybe not appropriate for this framework, but I can't tell.
Right. I don't think I have a good answer to it, but I think that the general answer is that the framework does require a signed estimate. in a lot of situations, the sign estimate is not really readily available. So I think that means positive and negative means different things, and then 0 means something. So if you understand, so you may translate that into your particular context. So the application I selected, because I'm familiar with those, and then so that fits this scenario. But I think the key thing is the DC criterion. So you need to measure, you need to come up with some sort of assigned effects to use this framework. And then I will be the first to acknowledge this is not universal to all problems, right?
Okay.
So if in some cases you cannot find a proper way to translate into assigned effects, then I don't see it's applicable.
Okay, cool. Thanks.
No problem. Thank you.
All right. Okay. Thank you again, William and Yi, and thank you for everyone who joined us today.
Thank you. Thank you, Jing.
Thank you.
All right.
Bye-bye.
Bye-bye.